We can see the detail in plant and animal cells through the use of a microscope. Now, microscopes started off really simple, and this was a fair few years ago now, but there have been massive developments, and modern microscopes are very complicated. We can see in much more detail and clarity. And which is why we now know the wide range of structures that are inside a plant and an animal cell. And then our bacterial cell. And again, the inner one is the cell membrane. The outer one is the cell wall. In the middle we have our cytoplasm, and no nucleus, but we have our DNA kind of floating around in the middle there. They love asking about structure and function of the different parts of cells, so make sure you learn this bit really well. We have our very small, very simple yeast cell, but astonishingly important. It has some similar features to plant cells, like the cell wall, and then on the inside the cell membrane. It has the cytoplasm and a nucleus. You need to know the structure of animal cells and what all the different bits do. So this big blob in the middle here, this is the nucleus. That is where the DNA or the genetic information is going to be held, and this is in charge of that activities. Then we have these smaller blobs. These are the ribosomes. These make proteins. These squiggly bits are the mitochondria. And they are in charge of releasing energy, and this is where respiration happens. Tucking this all in, going all the way around the outside, we have the cell membrane, and this just holds things in. And it also controls in and out. So control stuff that goes in and then control stuff that goes out. And then all around the kind of like gooey stuff that is left, this is the cytoplasm. And this is just where stuff happens. It contains a lot of enzymes, and it's kind of just there. Plant cells have quite a few things that animal cells don't have, but if we start with similar things, so we have the nucleus, and again that is where all the DNA is held. Here we have the mitochondria. This is again where respiration takes place, where energy is released from, the ribosomes. Again, these are in charge of making proteins, and then this inner one here is the cell membrane, again in charge of what goes in, what goes out. Now the other things that they have are on the outside, so the outer one, this is the cell wall, and this is very hard in comparison, very rigid, and this is what gives it its shape. So it keeps the shape really well. We have chloroplasts. These are for photosynthesis. We have this big gap in the middle. This is the vacuole. And not a lot happens here. It just contains a very weak solution of sugar and salts. We are actually very, very complicated beings. So here we have a cell. Inside our cell we have our nucleus. Inside our nucleus we have chromosomes. I'll just put that arrow there. Inside the chromosomes we have all of this coiled up DNA. And the DNA is made of 2 strands. So this is our double helix, our DNA. And DNA is made up of, as we can see here, bases which are A, T, C and G. And the structure of this, the sentence that it spells, we can then go from DNA. DNA has sections in it which are a gene. You can think of that as like a word or a sentence in a book, and then the sections of DNA will code for a protein. And the proteins are the building blocks of everything. If we want to turn our strand of DNA into a protein, there are several steps that we need to go through. The first thing we need to do is produce a complementary mRNA. And that is messenger RNA. We need to take that mRNA and attach it to the ribosome. We then need to code all of the triplets into amino acids. We need to transfer the amino acids to the ribosome by tRNA. And then we need to link them all together to form our protein. Now each protein is going to be very, very specific. It's going to have its own sequence of amino acids. You're going to get lots and lots of different shapes, lots and lots of different functions. These are going to include things like enzyme, which have a very, very specific shape. If an enzyme falls out of shape, if it gets denatured, it's not going to work anymore. And because these are so specific, if we get a change in the DNA, a mutation, this can be really, really harmful because it can either affect the way the protein folds, so it can affect its function, or it can stop it being produced altogether. Proteins are long chains of amino acids. And amino acids are like the building blocks, the bricks of absolutely everything. And proteins go on and do loads and loads of really, really useful functions. So they build up the bulk of everything. So they are your muscles, all the bits in tissue that need building up, that's what they do. They are your hormones. They are antibodies. And they are your enzymes. And the enzymes are incredibly busy going around your body doing loads and loads of things. So what is an enzyme? Well, an enzyme is just a biological catalyst. Which means it speeds up a reaction. But stays the same at the beginning as at the end, so it doesn't change or get used up. Enzymes work by what's called a lock and key mechanism. Only one thing is going to fit into the space. So here we have 3 substrates, and you can see substrate number 1 is going to come along, it's going to try and fit in there, but it doesn't fit in there, so nothing is going to happen. So this one is just going to go off now. So substrate number 2 is going to fit in there, can't fit that way, turning around, can't fit in that way either, so that one's not going to work. Substrate number 3 comes along and shock horror, it fits in absolutely perfectly. So now we're going to have a reaction taking place. So now the substrate's fitted in, the enzyme can work its enzyme magic and actually do something, either break something down or put things together. But if the substrate doesn't fit in there, nothing is going to happen. It's only if the substrate fits in perfectly will something happen, which is why it's called a lock and key mechanism. Enzymes have an optimum temperature that they like to work out. Now in our body, that is 37 degrees, but you can get extremophiles, which like to live at really, really hot or really, really cold conditions. Now below that optimum temperature, they're going to work, but they're not going to work very well. They're going to be slow, they're going to be lethargic. And they get to that optimum temperature, they're going to work really, really well. And then very quickly, they're going to go down until this point here at which the enzyme is denatured. We don't say that an enzyme has been killed, we say it has been denatured. Say it is killed suggests that it was alive, but all that's happened is changing shape. The bonds that hold it all together have been broken, which means it's not going to work, so it has been denatured. The same idea applies when we're talking about pH, but an enzyme can be denatured by too low or too high pH. So we actually have this very narrow looking curve where either side of it the enzymes don't work, but then in the middle up here we have our optimum. Now, meiosis is something different. This happens in sexual reproduction, so it's going to happen in our gametes. This is going to be our sperm or our egg cells. And what we end up with is 4 different haploid cells. Haploid means it has half the amount of DNA. So it is going to have 23 chromosomes, not the 46 chromosomes or the 23 pairs that you would normally see in a skin cell, for example. Now, meiosis, you're only going to get 1 set of chromosomes, you're going to get 4 new cells, and remember this happens in your reproductive organs only. I have a really stupid, silly way of remembering the difference between meiosis and mitosis, and I've made a separate video explaining that for you. Mitosis is a process of a cell reproducing itself to form 2 identical cells. So this is the type of cell division that will take place in your skin, that will take place in your hair, when your body is repairing itself, or in asexual reproduction. And what we end up with, what we start with rather, is just one cell which has the DNA and the nucleus in it. It gets a signal that it needs to divide, and all of the chromosomes line up down the middle. Once they're all lined up down the middle, once they're ready to divide, they start cell division. Once they know that each half of the chromosomes is around each end of the new cell, then we can have new membranes forming. And then the cells can start to separate. So we end up with two identical cells that are made from one cell. So the definition of diffusion, because this is important, they could ask this as a keyword, is spreading out of particles from places where there is a high concentration to places where there is a low concentration. So what this means in air, if we start off with a high concentration of purple particles down here, they are going to want to spread out to this area of low purple particle concentration over here, so that they are all nice and neatly spread out. If we have a cell membrane involved, again, the purple particles are going to be the ones that want to move, and what they want to make sure is that there are an even number of particles on each side of the membrane. So we have an equal concentration, so things have moved from a high concentration to a low concentration. Osmosis is a special type of diffusion because osmosis is a movement of water. And that is the movement across a partially permeable membrane. From a region of high concentration to a region of low concentration. Now remember, with diffusion, we were talking about things that weren't water moving across, but osmosis is a special type of diffusion. This is where the water molecules move across. So water might be moving from an area of low sucrose concentration to an area of high sucrose concentration because the sucrose is the bit that can't move. So to balance out the concentration thing, it's the water that does the movement, and this is where osmosis takes place. Whereas osmosis is a passive movement of things across a membrane, active transport is the active movement of things, and this is going against a concentration gradient. So because we're going against a concentration gradient, we need to use energy. This could be something where we're absorbing nutrients from the gut and it needs to go into the blood. So the gut will probably be at a lower concentration and the blood will be at a high concentration, so we need to use active transport so we can uptake all of the yummy goodness from the gut into the blood. As the population of the world increases, so does the amount of food that we need to eat. And this is where one of the arguments for being a vegetarian comes in, because we have the sun, our lovely green sun here, and this is providing energy to things. Now, the energy will go into the plants, and if we eat that straight away, there isn't going to be a massive amount of energy lost. Whereas if the cows eat the plants, and then we eat the cows, you're actually going to get more energy loss. So it's not a very efficient way of doing it. So there is several excellent videos about how becoming a vegetarian can actually save the planet. Photosynthesis is the method that plants use to make food. They take in carbon dioxide, and water and convert it into glucose and oxygen. Now, for this to happen, they need light. Now, the reason we write it above the arrow like that is because we need it to happen, but it's not actually a reactant, so it doesn't get used up in the equation. Now, there are several different things that a plant needs to photosynthesis to take place, and we can call these limiting factors. So the limiting factors for photosynthesis are going to be light. So if we don't have light, we're not going to get a lot of photosynthesis, so at night. The reactants. For example, if we don't have any carbon dioxide or any water, then we are not going to get photosynthesis taking place. And the other thing is also going to be temperature. When we're looking at the rate of photosynthesis and light intensity, from a point where there's no light, there's very little photosynthesis or no photosynthesis, it is going to increase steadily up to a point where it is no longer the limiting factor, where something like carbon dioxide, water or temperature is then going to be the limiting factor. When we're looking at how carbon dioxide affects the rate of photosynthesis, it is going to go up steadily and then level off when something else, like light, becomes the limiting factor. The graph for how temperature affects photosynthesis is quite different from how carbon dioxide or light intensity affects photosynthesis. When we have quite a low temperature, we have a slow but steadily increasing rate until we get to the optimum of the peak temperature. After this, it is going to decrease really quite rapidly until we get to a point which is about 45 degrees, where the enzymes are going to be destroyed. And once this happened, once the enzymes have been denatured, once they've been ruined, they cannot function anymore. So there is an optimum temperature for photosynthesis. Below that, things will just work slowly. Above that, things just won't work at all. The glucose that plants make in photosynthesis is used for many different things. Now, believe it or not, as well as photosynthesis, plants actually undergo respiration. This is the same equation, just in the reverse direction. They also use the glucose for building structures, so like cell walls. And they use it for making proteins. This glucose can be stored in two different places. It can be stored as oil, so that we get out of the crushing of the seeds of the plants, and this is where it's going to be stored as a lipid, or it can be stored as a carbohydrate, so it can be stored as starch. And these are going to be things like your big fat potato tubers, the roots, the stems, the leaves, the seeds, loads and loads of different places that starch can actually be stored in plants. Aerobic respiration takes place when there's lots of oxygen around. So we have glucose plus oxygen will turn into carbon dioxide plus water, and most importantly, energy. Now I've put energy in here, even though I'm gonna put it in a little circle, because it's not really a reactant, it's just something that comes out. The reason we do aerobic respiration is to get energy out of things, but it's not really a reactant, so the chemistry teacher in me doesn't really like putting it over there. Now, this aerobic respiration, it's going to take place in your mitochondria. And like I said, this is only going to take place where there is lots and lots of oxygen around. If we don't have a lot of oxygen around, we have anaerobic respiration. And the glucose will then turn into lactic acid and the energy again. Now the lactic acid, you're going to get this when you're doing things like sprinting, running really fast, doing lots and lots of exercise. That's the stuff that hurts. It really, really hurts when you do loads and loads of exercise. That is because once you've stopped, you've built up this oxygen debt. And you have to breathe really heavily because you have to repay this oxygen debt. So blood's going to be flowing around. It's going to want to be removing the lactic acid and turning it into carbon dioxide. One example of an organ system is the digestive system, where we have lots of different tissues working together, lots of different organs all working together to perform one overall function. So we have the large intestine, which reabsorbs water. We have the small intestine, where food and sugar and various different things like that are reabsorbed. We have the stomach, where the food is digested. We have the liver, which adds in things like bile, which helps to neutralise the stomach acid, and then we have the mouth, which breaks down the food. So lots of different organs, the liver, the stomach, the large intestine, all working together in an organ system. The digestive system is packed full of useful things that do stuff. So the first one that we come across is amylase. And amylase will turn starch into sugar. And this is found in a number of places. It can be found in the saliva, which is why if you put a bit of bread into your mouth and just leave it there, it will eventually, after a while, quite a long while, start to taste sweet. That's because the amylase in your mouth is just breaking it down. It comes from the pancreas and in the small intestine. The next one that we come across are proteases. And they will take a protein and break it down into amino acids. Again, these are found in lots of different places. So the stomach, again the pancreas, and the small intestine. Next one that we have are lipases, and they will break down lipids, which are fats, and they will break them down into fatty acids and glycerol. These can be found in the pancreas and the small intestine. And then lastly on this slide, I'm just going to mention bile, because it's not an enzyme, but it is involved in this. And what it does is it's just two things. It neutralizes stomach pH, so the hydrochloric acid in the stomach is way too acidic for enzymes in the small intestine to work properly. So you add the bile in once the hydrochloric acid and the digestive goo has come out of the stomach, so that the small intestine is a bit more alkaline so it can work properly. The other thing that it does is that it emulsifies fats. Which means breaks them down into smaller lumps so it has a larger surface area. And the bile is produced in the liver and stored in the gallbladder. One place active transport happens is in the gut. So here we have our small intestine. And the really interesting thing about the structure of the small intestine is that it's not flat, it's not smooth, it's got all these little wiggly bits, and these little wiggly bits are villi or villus, and these have a large surface area. And the reason they have this large surface area and this very thin wall is so that they can absorb as many nutrients as the blood from the gut into the blood as they can at once. Now this can either happen by active transport or it can happen by diffusion, depending on what is being absorbed. In the lungs, we have the gas exchange going on. We breathe in the oxygen, and that needs to transfer from our lungs into our bloodstream, and we breathe out carbon dioxide. So we need to transfer the carbon dioxide from our bloodstream into the lungs so that we can breathe it out. Now, what we have in the lungs are these little alveolus or alveoli, which have a large surface area. And surrounding them, they have this network of capillaries, which increase the rate that the oxygen and the carbon dioxide can be transferred across. And now this is going to be diffusion of oxygen and carbon dioxide as they move from a high concentration to a low concentration. In the respiratory system, we have our esophagus, which is part of the where we take in food. That goes down here to the stomach, and then we have our trachea, which goes into our lungs. These are our two lungs here, these two very large organs, and then at the bottom here we have the diaphragm. Surrounding this, all around the outside, we are going to have the rib cage, and then in between the rib cage and the lungs we are going to have the intercostal muscle, and then sitting in here we're going to have the heart. And now the tubes that come from the trachea and actually feed off into the parts of the lungs, this is the bronchus. Here we have them one on each side. And when they split off even more, they're called the bronchioli. And these lead up to these alveoli here, which are these like bunches of grapes where the gas exchange actually happens. Now, if we think about what ventilation actually is, this is the movement of air. And if we want to breathe in, what we need to have is a flat diaphragm. We need to have our intercostal and our ribcage muscles going up and out. And these two things will drag air in. Now, if we want to breathe out, we need to have the opposite happening. So our diaphragm is going to move up, and then our rib cage is going to drop down. And this is going to send air out. Now, when we are looking at an area, when we want to study this distribution, we can work out what lives there in a couple of different ways. And the first way we can do it is with quadrats. So what you need to do is to get your quadrat and place it randomly. So basically chuck it in a field somewhere. You are then going to need to count everything in the square. And because we're scientists, we don't just do one thing, we do this 3 times. So we are going to need to repeat our counting. Once you've repeated our counting, we need to work out a mean, and then we need to move the quadrat to a different location within the area. Again, we're going to need to count stuff, repeat the count, work out an average, and then once we've done that, we can compare the distributions. When we're talking about where a living organism lives, there are a couple of words that we use. We use habitat. That is just the place that it lives in. And we use distribution. So that is where it's found within that particular location. And there are going to be a number of things that are going to affect distribution, whether it's distribution of the grass, whether it's distribution of the trees, or whether it's distribution of the animals in there. And these things are going to be temperature, the amount of water, oxygen, carbon dioxide, nutrients, and light. Because whether you're an ape, whether you're grass, you're going to need to be near somewhere that has water. You're going to need to be somewhere that has the right levels of oxygen, right levels of carbon dioxide, where you can get food from, where you can get light, and where it's not too hot or not too cold for you. Well done for making it to the end of the video. That was quite a long one there. Good luck for your exams. If there's anything I can do to help, just let me know.